And clear.
Excellent.
Can you try sharing your screen?
Yeah, let me set it up in a way that I can see it in two screens. And
Perché sennò tutto quello che diciamo viene detto qua. Ok. Sennò perché non abbiamo parlato. Così c'è solo il recording, qua c'è scritto solo il recording, ok, e la trascrizione basta. E quella che ho scritto? Cioè la roba che ho registrato per quanto tempo dov'è? Adesso dovrebbe arrivare il link. Perché non c'è ancora? Ah, questo è il trascritto, non è il chat. Ok. Questa 10 secondi. Quindi stop recording, stop trascrizione. Esatto.
Sì. Beh, è stato, non posso fare una parte più minuti.
Non è arrivato.
Antton?
Sì.
Antton?
good morning.
Okay, the connection via Teams.
Sorry, can you repeat that?
It's good, the connection of the Teams.
I cannot hear you properly. The sound is a bit cranky.
Okay, Antton? Okay.
Now it's better.
The connection is Teams, the voice and the video, okay?
Yep, now I can hear you properly, and I can see the classroom, and I can see a screen as well with the slides.
Ok, le slide che sono le sue,
Sì.
per far vedere il calendario,
Delle lezioni.
settimana.
Lui sta condividendo adesso
Sì.
il calendario.
No, facciamo doppia connessione.
Questa non si può aprire da qui.
Il calendario.
Sì.
Lo vedono in sala?
No, adesso no. Sì, lo vedo in sala. Ah,
E lui lo vede?
allora, lui no, perché sta condividendo
va bene
lo schermo.
E
Poi io
chi è collegato non lo vede. Chi è in sala vede la settimana,
Sì,
ma chi
sì.
è collegato dei studenti nessuno.
Sì, ma loro devono vedere lui.
No,
Sì, sì, ma poi lo togliamo, cioè forse era solo
Dopo?
all'inizio. Sì, sì.
Ok, va bene.
Cioè così
No,
vedono questo.
Sì, sì. Ok.
Adesso vediamo il calendario.
Ma c'è un modo poi per mettere in grande, il super grande il suo schermo per far vedere le slide? Forse quando le condivide lui, sì. Cioè
Sì,
quando
sì, quando
allarga
le
lui.
condivide lui, sì.
Ok, ok.
Meglio
così.
così. Voglio qua. Ecco.
Così.
Allora. Noi dobbiamo mettere in focus. View. Speaker.
Ok.
Focus on content. Non ci vede neanche lui, capito?
Ok. E se vuoi tornare indietro? Ah, hai rischiacciato sugli stessi.
Sì, sì.
Ok. Però tanto secondo me qua lui quando condivide si vede solo lo schermo. No?
Lui? Lui sì. Forse vede anche noi, Mauro. Se riesci a toglierti la camera.
Sì.
Perché così lasciamo. Lui vede qua e noi vediamo lui.
Ok.
Allora così.
E poi metto comunque questo, speaker e focus on content.
Se metti focus on content non vediamo
Non vediamo
neanche
lui. Ok, allora lasciamo così.
Quindi forse è meglio lasciare così.
Ok.
Adesso tutto a posto adesso.
Devo Continuando la presentazione.
La tua. Gli altri sono tutti a posto con la nota, perché tu mi hai insegnato camminare.
Sì, ok.
Quello fatto a posto.
Ok, dovrebbero essere
Sì,
tutti
sì,
a
a
posto.
posto gli altri.
Sì.
Quindi serve soltanto per chi non è di volume. Che sono io controfirmi. La tesi io devo controfirmare.
Devo confermare l'ambiente perché sei il mio relatore.
Poi la famiglia uguale che francese e nato pure perché diciamo che siamo
Quelle univo, bastava quelle univo.
allora sarebbe
Professor, 5 more minutes and we'll start.
siamo entrati se devo mancare soltanto la gente 5 minuti ci riprendiamo
Sì, ho chiesto.
a Antton.
Sì. Se vuoi dirlo a lui?
Sì. Antton?
Yes.
5-10 minutes for organize the student in the hall.
Sure, no problem. Just let me know when you're ready.
Ti posso per la serie? Solo sentire.
No, sono sfinita. Abbiamo fatto un evento questo weekend, quindi da lunedì scorso che lavoro senza smettere.
Non ci mettiamo su quella riflessione. Non è male, ma
Professor,
siamo tutti? Siamo tutti Giada? Giada?
Greta.
Greta. Non ci va adesso. Siamo tutti? Iniziamo, quindi.
Non è la prima volta che
Con le distanze.
che lo sa fare. Anzi. Tanto c'è di quelli che sono dei canalisti che l'hanno già fatto.
Va bene, tutti.
Sì.
Iniziamo. Che apre la porta. Sente, good morning, everybody. Welcome to Fano. Welcome to the teaching week per il 40° ciclo del FIS Teaching Week e per il 38° the end of the last teaching week. The new scambio of. In this teaching week, good morning for Jeff. Jeff lo conoscete, is the academic board from Holland. È arrivato. and Yvonne, the new PhD student from Columbia in 40º ciclo. This is the Settimana Teaching Week. In the morning, Antton from Denmark. In the afternoon, the Frederick in presence in all for start this day. Antton, the teaching with Ologenomics applied to host microbiota interaction in marine organisms. Important for the new administrative question for the credit. Okay, this is discussed the problem for the 40º is necessary. And at the end of each lesson, the professor describe the modality of the interaction for the briefing. Okay. Each teaching is adopt one method different for each problem. Okay, good job, Antton. Anton. Okay.
Thank you so much. Yeah. Can you hear me?
Sì?
Yes.
Sì.
Yes.
Siamo tutti, meno che la sala. La sala.
Just let me know when.
Okay, Anton. Thank you. Please.
Thanks. Good. The connection, so I think the sound is not that good. I hope you can hear me properly. Let me know otherwise. I hope it's all fine. But I cannot hear you right now. So
Yeah, it's all good.
all good. Okay, thanks. Thanks a lot. Well, thanks a lot for the introduction. And so my name is Antton. I'm from the University of Copenhagen, and it's a pity I cannot be there, but due to family reasons and other traveling, etc., it is quite complicated for me to travel too often. But I will try to give this lecture this morning in the most interactive way as possible. I know it is not easy because you're there, I am here, and it's been already a while since COVID times, and I guess, well, at least me, I've lost the routine of engaging students through applications, etc. But I will try to do that again. So we will be using Slido. Hopefully it will work. And if you have your mobile phones at hand and you can then scan the QR code, etc., we will try to interact this way. Okay, so hopefully it works. If it doesn't work, then we can do it in a more traditional way. I don't know whether there is a microphone there and then the students can talk directly, or can, I don't know how this might work otherwise. But well, that's not a problem for now.
We can try with the microphone later.
Okay, well, hopefully it's not needed, and yeah, let's try make this work then. Okay, so just a little bit about me and our center and what we do. So I belong to the Center for Evolutionary Hologenomics, which is a research center within the Globe Institute, within the University of Copenhagen. So we are placed in the very center of the city of Copenhagen. And what we do is to try and understand how microbes, mainly bacteria, and animals, mainly vertebrates, have affected and are affecting the evolutionary and ecological processes, both sides. So how microbes affect animals and how animals affect microbes, considering their ecological interactions in the environment and also the evolutionary processes they undergo. So we do all kind of research in terms of experimental work and field work, a lot of genomic work. And today I will try to give you an overview of what we do and what the field of ologenomics is. I know it might be too early, so initially this lecture was not scheduled as the first one in the week, so it might be a little bit tough, but hopefully, yeah, hopefully you like it, and I will try to make it as light as possible. But here within the Center of for Evolutionary Hologenomics, we belong to what we call the Albderdi lab, and we are currently about 20 people that are working on this animal microbiota interactions. We are biologists, chemists, engineers, mathematicians. So we are a little bit of a mixture of people. And there is also a lot of people that come here for short visits, etc. This is a photo we took the last summer, but I would say almost 1/3 of the people there are not in the group anymore. And new people have come, so it is very dynamic. And you are also very welcome anytime to come and visit us. Usually Copenhagen, it's not as sunny as in this picture, so we were very lucky last June, but that's how it is. So how I have structured the lectures for this morning, there will be 4 sections and sessions. Each approximately will last 1 hour or 55 minutes, and then we will have some breaks. This is a problem, so don't actually pay attention to the minutes up there. So this was from an old slide that I forgot to change. But we will have the first session until 10:30 more or less, then the next one 11:30, 12:30, and until 1:30. And we will have at least gaps of 5 minutes, probably 10 minutes. Some of the sessions might be longer than others, but then we will try to adjust as much as we can to this schedule. So this is the plan for today. And then, as it was mentioned before, this year, you need to hand over an assignment that I will be assessing in May. And what I will ask you this year is to create a short grant proposal on a project that deals with hologenomics. So we will see what hologenomics is, and we will touch upon different aspects of doing research using hologenomic methods. And then using all this information and all this knowledge you gather today, you will need to create your own fake grant proposal, but that hopefully also it might be useful for you in the future. So it's gonna last like 3 to 4 pages, and the structure will be fixed. You can so the amount of characters that I have set here is more or less just as a guide, but the structure that you should follow is this one. So imagine that there is a funding agency that is requesting you a grant application that is about 3 to 4 pages long. And this is the structure that this grant proposal needs to have. It needs to have an initial background or state of the art explaining what is that we know about the system, about the question that you want to address, followed by some hypotheses and objectives that you want to address in your project. Then there will be a chunk for the methodology, which methods, which study design you are going to use, and then the practicalities and how you are going to run the actual experiment and the study. So how are you going to structure? How many people are you going to hire for running the project? What's going to be the budget of the project, et cetera, et cetera. And then in the end, there will be a final section about the impact that will deal more with the societal aspects and the broader impact that your research is expected to have, finishing with some references. So something like 15, 20 references. For the backgrounds, what I would like to ask you is that you start from the big picture. So whenever you have an idea, start from the big picture and then try to narrow down to your study questions. So provide enough information to contextualize the hypothesis and objectives, and then avoid as much as you can any kind of cluttering information that does not contribute to the application, so to your grant. So this should be more or less the structure of the background, which is a funnel-like structure that you start trying to engage the evaluator with the importance, the relevance of your project, and then taking that evaluator to what you want that person to be, to understand your objectives and your hypothesis. The hypothesis and objectives should ensure that the background information is good enough to make them understandable for a general reader or general assessor. Let's imagine that the people that will assess your project are not expert in this topic, so you will need to explain the background properly so that the hypothesis and the objectives are quite self-explanatory. And then you will also try to make this hypothesis as addressable as possible with the proposed methodology and study design. So based on the knowledge that you will obtain, hopefully this morning, then you will need to make this effort and say, okay, if I have this question, if I would like to address this question, then I will need to use this methodology and using this study design. And then the evaluator will need to assess this properly. The evaluator in this case is going to be me. In the methodology, you will need to explain about the study system. Are you using a mollusk, a fish, wild animal, a farm animal, etc., etc. What kind of, it's going to be an experimental work, it's going to be a field work. How many samples are you going to take? How are you going to process them in the laboratory? What kind of bioinformatics analysis you will need to do? What kind of statistics will you do to address or to test the hypothesis you have set out? So all this will need to be explained in the methodology. Briefly, you won't be able to give a lot of details, but try to be as concise as possible to make the entire structure as coherent as possible. And then in the work plan, what I would like to ask you is to make an effort to structure in time your project, what is going to happen first, what's going to happen then, and try to assess how long each task is going to take and what type of organization of these tasks will exist and which people you will put in charge of running those tasks within your project. It is very open in the sense that you can aim for a project that it is just, let's say, 50,000 euros of budget, or it might be 10 million euros of budget. You decide. And but whatever you write should be adjusted to that, to those numbers. It can be also a 1-year project. It can be a 3-year project. It can be a 10-year project. So it's up to you, but try to make it coherent as possible. And then in the final part, I would also like to ask you to write a little bit about the impact. So on the one hand, what is the scientific impact of the research you are proposing? So how is it expected to contribute to the scientific community? How do you expect that this will change your field, etc.? And then also, what is the broader societal impact that you expect for your results in terms of sustainability, biodiversity, environment? It can be also the economic impact. If you expect that what the experiment you are going to do, it's going to produce some kind of tool or some kind of resource that will have a commercial impact, you can also mention that in this section. And finally, a small section on references. As long as you can, use reliable databases and prioritize peer-reviewed articles. And just for your own sake, if you use reference managers like Mendeley or Paperpile or whatever else that you want to use, it's going to be easier for you. But this is what the assignments will be. We will get back to this in the end of the lecture, but I want to give you this in front so that while we go through the different lectures, you can also gather the information and you can start already thinking about ideas of what you would like to work about in this project. I would also advise you to do this as soon as you can and without leaving it for May the 8th, because you are going to forget a lot of the things that we explained today. But then what you will need to do is to go to this link here. So rather than sending these messages or these works to me directly through email, what I would like to ask you is to use this form to hand your assignments over me. And if you use this, I hope you can see it. If you click here in this link, you will get into this form. I hope you can see that. So please add your student name and surname, your email, then the title that you are giving to your grant proposal, and then attach your file here. Okay, so click, and then you can just drop the file here, or you can browse it in your computer, and then you will get an email, automatic email saying that this has been sent properly. This is a way to ensure that all the works gets stored in a database and they are not lost in my mailbox, and then we might have problems. So please, if you have any questions, you are very welcome to write emails to me. But for handing over the work, please use this formulary so that everything is stored properly, and then I can ensure that I can evaluate all the works. So, do you have any questions on this? If you do, perhaps you can raise your hand, and I hope someone can give you a mic, or you can write it directly in the chat, as you prefer.
No questions.
No questions. Excellent. So then we can start. So, as I mentioned, there will be 4 sessions, each 1 hour more or less. And the first one is just introduction to hologenomics. What do we mean by hologenomics? What is this about? And so we will do the first Slido attempt here. This 2 Antons, it's me trying to validate this is working. So can you please try join the Slido app? And you can use it. You can use your mobile phone to scan the QR code, or you can go to slido.com and then use this this code. This is going to be a short one, an easy one, but it is mainly the validation to see whether it is working. Good. Lucia is already in. I don't know how many students are we expecting today.
36.
36. Okay. So.
No. No.
Should expect 38 then. It's...
I know. Aspetta. Sì.
And I have prepared a couple of questions that I would like you to answer. They are easy to respond. So whether the response is correct or not, that's a different thing, but it's a yes, no question. So let's see if the rest of the people can join. We'll try to get at least to 30 before going ahead.
Professor, just 1 second. Is everybody connected to the internet? So you can access Alma Wi-Fi and enter your Unibo credentials. So the Studio Unibo one will suffice.
At least 29 people managed to get in. Let's wait 1 more minute. And then there will be a few more interactions. And if this is working, I think this is going to be better for the lecture. So let's make sure that as many people as possible are able to join.
Are there 31 people on?
33.
So 2 is me, so there are 31 now.
Okay, so we're missing 2 people. Pero c'è uno che... There's somebody that's at home. They're sick.
Mm-hmm.
Io 32.
34. Sono...
Non riesco a contare.
Là sono 5.
5,
5. Là sono altri 5.
3, 7, 19, 22, 25, 26, 27, 29, 30, 31, 30, 33.
Ho io quanti sono qui? Adesso 34. Metto anch'io.
No, ci sei entrato anche tu?
No.
Sì.
Ah sì?
Quindi sono...
34 minuti.
No, quindi sono 31. No, però 2.
Ma una manca perché non è arrivato.
Just let me know if you feel that we should go ahead and.
1 more minute, if that's okay.
Yeah, it's completely fine with me. This is the trickiest one, the first one. Then it's gonna be easy for the following ones.
Non l'ho contato. Questi 36 sono già senza
Bisogna vedere chi li contatti.
Non è giusto. No, ne manca solo uno. Ne manca solo uno.
Now I just hope that when I click, it works, because it's gonna be a pity if it
Okay, I guess we can proceed.
Okay, let's give it a go. Let's see what happens. Okay, so you have the first question here. You have 20 seconds to answer yes or no. Do all animals have an associated microbiome? Here are the results. So 23 out of 34 were able to answer this within 20 seconds. So the next one, you will need to be a bit faster if you can. But most of the people, it was quite overwhelmingly yes, that most of you think that actually it's probably everyone but one person, that all animals have an associated microbiome. Good, let's go to the second question. Are animals able to detect and react against all microbes?
No.
Okay, here the results are quite different. So 1/3 said that yes, and 2/3 said that no. So I won't give you the answers of these two questions now. So we will enter into, with these two ideas, we will enter into the lecture. And this is, so all the questions were right. So the fastest one is the winner here. So congratulations for being fast, Giorgio. Perhaps the response were not correct, but you were fast at least. Cool. So you mentioned that, almost everyone mentioned that all animals have an associated microbiome. The truth is that there is a gradient. There is a huge spectrum of possibilities from some animals to others. It's true that all animals live in an environment that is surrounded by microbes. Then the fact is, so the important fact is how we describe or how we define what is being associated to a host. Microbes are everywhere, but whether a microbe is associated to a host, then it might be a different thing. And this association can be defined in very different ways. We can say, okay, perhaps we believe that just being touching a skin inside an intestine might be enough to be associated. Some people might argue, though, that there needs to be some time components considered, that if a microbe is attached to someone for a short time frame, perhaps that doesn't mean that it's associated. But if it is for a long time, then it is associated. We could also consider that perhaps that's not enough either. So there needs to be some kind of interaction. The microbe needs to give something to the host, or the host needs to give something to the microbe in order to consider this associated. So the actual definition of what is to have a microbe or a community of microbes associated with a host is not that clear, and it is very question specific and system specific. I think, just a small parenthesis, I think I am hearing myself with some kind of delay, which is a little bit annoying. Is it possible, Francesca, to mute for now your microphone? Yeah, I think that's going to be better. And then if you have any questions, please so add it in the chat, and then it's going to be easier. But here in this figure of a manuscript that we published 3 years ago now, we exposed a gradient of possibilities, not in the degree of association, but in the degree of diversity in this case. And on the right side, you will see a vertebrate. It's a bird, a black bird there, with quite a lot of microbes, with hundreds of microbes. And in the other spectrum, we have a larvae of a lepidoptera, of a butterfly, where there is actually no detectable resident microbes. So there are some animals that, even though they might have microbes attached to them at a given time point, this is not happening recurrently. And if you sample different caterpillars, you might be getting quite different microbes. And these microbes are any kind of very ubiquitous microbes that you can also find in the leaves, in the ground, etc., etc. So it is true that although all animals are somehow linked to a range of microbes, they might not all be associated with microbes. And they might not be really getting from microbes some kind of advantage or some kind of change in their biology. In some cases, it's going to happen. In some cases, it doesn't happen that much. This said, even though there might be some cases in which microbes are not that relevant, overall, and in most cases, microbes play a huge effect on different processes, biological processes of animals, and not just animals, of animals and plants. So any kind of eukaryotes or larger organisms. And here you have 5 examples, starting from corals and dinoflagellates, following by termites and bacteria that contribute them to decaying the wood, to degrading the wood. We also have an example of crop plants, like could be maize, it could be wheat, and the microorganisms that are associated to the roots in the soil. Another example of a cow and the rumen microbiota, that it is the only way that ruminants can actually degrade the polysaccharides, the complex polysaccharides that they take from the grass. And these animals, for instance, if you remove the microbiome from the rumen, they just die. So they completely depend on microorganisms. And then here we have another example of a laboratory mice and their good microbiota, which is important. Perhaps the mice can live without a microbe or without a community of microorganisms, but their biology changes considerably. So in different contexts, starting from could be very fundamental natural processes like the effect that corals have in coral reefs, etc., to agriculture with ruminants and crop plants, also aquaculture, which is more relevant. And I apologize for not having that example that is more linked to the study program here. But there is a considerable impact of microorganisms on host animals and plants. How we analyze that is what it is different. So I would say for a long time, since the development of molecular methods, the main approach for understanding why the biology, why the phenotypes, why the behavior, why the physiology of animals was changing was the analysis of their genomic data equated with the environmental changes, environmental fluctuations. And that's what we see, or what I call it here, the genomic approach. So I'm pretty sure that you have seen this, this very simple equation that the phenotype equals the genotype and the environment, the interaction between the genotype and the environment. And this model has been used for a long time for improving production processes, for understanding ecological processes, evolutionary processes, etc., etc. I would say since 2000, 2005, more or less, the focus started to shift towards the microorganisms. And there was a lot of work that was done using this other model, which I call it here the metagenomic approach, where the phenotype of an animal could be a mouse, but could be a fish, could be any kind of mollusk, is understood as the interaction between the environment and the microorganisms that are associated with them. So P equals metagenome, mg, multiplied by the environment. What we argue in our center and the kind of research we do is that neither the genomic approach nor the metagenomic approach is good enough to understand the actual process that is happening here, because the host domain is not independent to the microbial domain, and the microbial domain is not independent to the host domain. So we need to consider both in order to get to a more accurate understanding of how the environment, the host genomics, and the microbial metagenomics are affecting the phenotypes. And this is fundamentally what we call hologenomics, which is analyzing jointly the animal genomic and the microbial metagenomic information to understand how animals are adapting to the environment, are responding to a treatment, etc., etc. How we do this is that we consider both the genomic domain and the metagenomic domain. Genomic is linked to the animals, metagenomic is linked to the microbes, and then we generate and analyze different omic layers to understand how the different structure of genomic information is being expressed and how this is actually changing the system that ultimately affects the host phenotype. And this is what we call it the holoomic approach, which is not just analyzing the genome and the metagenome, which gives us information about the potential of the animal genome or the plant genome and the microbial metagenome, but also how all this potential is actually expressed, it's actually realized, so that the final phenotype of the host in this case takes the shape that we observe. Why this is important, or why this is relevant, is because the hosts, the animals or the plants, and the microorganisms continuously interact. And this is something that I was asking in the second question, whether you think that animals are able to detect and react to the presence of bacteria, or bacterial microbes in general. So the question is that in most cases, yes. In some cases, no, because if we talk about bacteria, if we talk about archaea, if we talk about different kind of viruses, some are more detectable than others, and some do create some kind of processes that are also more detectable than others. And the capacity the hosts have to detect and to identify what type of microorganism is interacting with them is also different. In some cases, the microbes are able to identify at the almost strain level or species level to say, okay, I know what this guy is and I know how I need to react against this guy. In some other cases, that reaction is much broader. In some cases that I say, well, I know that this microbe has peptidoglycan, this bacteria has peptidoglycan in the cell wall, and I will react to the presence of peptidoglycan, but I don't really know what else is there. I'm just reacting to the presence of peptidoglycan. So sometimes it is very specific, sometimes it's far less specific, and sometimes there are some microbes that they are just ghost microbes. The animals or plants are not able even to detect that they are there, but they might be affecting the processes that are undergoing. And we will see some of those examples now. Here, the relevant thing is that when we talk about mainly the gut microbiota, which is probably what you all have heard about, it's not the only animal-associated microbial community that is relevant. So the skin microbiota can be also very relevant in certain contexts, but for most of the systemic functioning of animals, the gut microbiota is the most relevant one. And it is relevant because it is not only affecting the gut and the processes that are happening in the gut, but are also affecting a lot of other organs through different ways that the gut environment has to transfer information and to affect other organs. So we have the gut-brain axis, for which things that are happening in the gut can affect the brain and can affect behavior. We have the gut-liver axis, that it is much more linked to metabolism and how different type of compounds are going to be metabolized by the animal. We have also a gut-kidney axis that is more related to the excretory system, and the microbes in the intestine might be affecting how good or bad the animal might be in excreting compounds that needs to be taken out from the body. And we also have a gut-lung axis that is more important for terrestrial animals, that also affect how the animal's capacity for respiratory capacity, etc., changes. I was mentioning peptidoglycan before, and peptidoglycan is one of the compounds from the cell wall of some bacteria, but it is not the only structure that animals or plants are able to detect. We have DNA inside the cell, we have the ribosomes, we have the flagellum, we have many other structural components of the bacteria that animals, could be fish, could be mollusks, could be any other kind of animal, are able to detect. The more complex the animal it is, and the more complex usually the adaptive immune system is, the higher capacity will be to be more specific against any of those elements that can be detected. The way this is done from the animal perspective is that the intestinal cells, the epithelial cells, have different type of receptors. We can be talking about toll-like receptors that are in the cell wall, that these are in the cell membrane, that are exposed to the intestinal lumen, or node-like receptors that are inside the intestine, inside the cells, that are able to detect different type of microbial stimuli. These microbial stimuli can be flagellin, which is the main protein of the flagellum of the bacteria, but could be lipopolysaccharides like LPS that you can see here, could be DNA structures that can be also detected. The bacteria themselves can be producing toxins, can be also producing some kind of compounds like short-chain fatty acids, like uric acid, etc., that permeate the cell, and then the node-like receptors are able to identify that and trigger a response. And this response can be quite different depending on what kind of stimuli we have and what kind of receptor has been able to detect that stimuli. This MAMP stands for microbe-associated molecular pattern, and it refers to any kind of molecular component that animals or plants are able to detect, identify, and trigger a response. So once we have a receptor that has bind to a MAMP, that usually is linked to an adapter that amplifies a response. And this response can be either local or can be systemic. And usually the local response is related to the innate immune system, and we might be talking about the release of AMP, so antimicrobial peptides. It could be also different types of immunoglobulins that are released. But as I mentioned before, the gut is also related to other organs, and the response can be much more systemic. And usually that systemic response is more linked to the adaptive immune system that also creates a much larger and much more complex response to at the entire organism level. The main difficulty of complication that we have in this case is that we have microbes and we have hosts, and I will say that microbes produce some kind of molecular patterns that hosts can detect and can create a response, and this response affects microbes again. So we create a circular loop that it becomes extremely complex because there are always cross affecting the microbes and the hosts, and telling which one is affecting what is quite different because it is very difficult to stop one of these processes and tell what is the cause and what is the effect. So this is something that is quite important to consider when doing any kind of animal microbiota research, which are the interaction loops and the complications that derive from them. And we will be talking about this later and the problems and the complications they introduce. So this was the first part, the introduction, and that was, we are in time. So what I would like to do after every session is to ask you to, let's say, assess yourself how much of the explanations of these sections did you understand. So I would like to get a feeling of whether you've been completely lost, and then we might want to go back and touch upon some of the aspects that require some more explanation. And if we see that most of the people understood the main messages, etc., then we can just go ahead. So if you join Slido again, then we can assess where we are. And if you have any questions, depending on where we are, we will spend some more time and we can deepen into some of the questions. And just remember, it's not about how much you liked or disliked, but it's more about how much you feel you understood what I was explaining. And those who have some uncertainties of what you think that some of the aspects were not clear enough, please use the chat or raise your hand, and hopefully we can have some interaction in the following 5 minutes that we have before the break. And please don't be shy. So we will make this work. So if you have any questions, please, there must be some questions. Otherwise, it should be, the score should be 10 and the score is 7.8. So I will stay here. I will endure here until I hear some questions. And I can be very patient, and it can become very awkward for everyone. So please ask a question. I cannot give a candy to the first one that asks a question, but I guess Francesca, Mauro, someone else can. So.
No questions.
No questions.
No.
Too shy. So I hope that in the next one, it's going to be much more complex than the other one. So if the score is at this level, then please ask questions for your own sake. Remember that you need to deliver then an assignment. Okay, but I can see you are quite shy. So shall we have a 5-minute break now? And then we start with the next session at 10:25.
Okay.
It'll be a bit longer. Yep.
Okay, thank you.
So I'm back. Whenever you're ready, let me know, please.
Just a second, Professor. There's a bit of a line for coffee. Okay, Professor, we can continue. Thank you. We can hear you.
Thank you. Yeah, my mic was up. So I was saying that the second session is an experimental design for hologenomics. So I think this is going to be more relevant for the specifics of the assignments you will need to send to me. So it will have three main sections, and we will start from the general consideration of different experimental designs related to the complexity of the system that we are analyzing. So pay attention and try to link it to the kind of system you usually work, and also link it to the kind of questions you might be able to answer using that system. And in order to do that, let's see if I can make this work. Yeah, so let's go back to that initial equation that I mentioned in the previous session, where we don't try to predict a phenotype by equating the genome and the environment or the metagenome of the environment, but we try to do it all together instead. So we have 3 dimensions that we need to consider whenever we are trying to use hologenomics to understand some kind of outputs in an animal host. The genome of the animal, the metagenome of the microorganisms that are associated with that animal, and the environmental factors. And most importantly, the interactions between them. So we need to always consider that all these aspects might be interacting with each other, and whatever change that is happening in one of the variables might be affecting the other one, which is a source of complexity that we also need to consider. So in this publication that I mentioned before, we contextualized this as a 3-axis volume, a volume with 3 different axes. Here you can see the first axis is the genomic complexity, then we have the metagenomic complexity, and then we have the environmental complexity. And what you have, what you can see here is a conceptual visualization of how complex the volume that these 3 axes would take is depending on what kind of system we are working with. So we have the wild species that you can see there that have the wider volume in this 3-dimensional axis. So you need to make this abstraction to see how this is a cube actually. Then we have some wild populations that are within a species that are still complex, but are less complex. And if we go narrowing down, we end up with some inbred germ-free models that have very low genome complexity, low metagenomic complexity, and as this is run in a lab environment, also a very low environmental complexity. So what we argue is that depending on what is the amount of complexity that you have in these 3 different axes, the kind of questions you can answer is different, because you might be unable to control a lot of things that you need to control in order to answer some questions. Let's go step by step and try to take each of these levels to your own reality in your PhD projects, in any kind of research or research environments you are involved in. And let's start from the most complex systems. So the most complex will be the wild species in a wild environment. So if we are trying to understand how the animal microbiota interactions are happening in the wild, using some corals in the wild, or some fish, or some jellyfish, or whatever, we need to consider that, and we are comparing species to each other, we need to consider that the genomic information that we have there might be very complex. The different species will have different genomes, the different individual within a species will have different genetic differences at the population or individual level. We also need to consider that as we are not controlling the microbiome, then the metagenomic complexity can be also very large. And the environmental complexity, which is something that is also outside of our control, it's very large. This means that we might be able to answer some questions, but we might be unable to answer a lot of other questions because we don't have enough resolution to control the different elements that are interacting with each other. And there will be so many things that we are missing that will enable us, that will prevent us from addressing some of the questions. This can be narrowed down a little bit if we specify in a single species. So the genomic complexity will be slightly lower because they all belong to the same species. But still, if we are working in the wild, it's going to have a lot of metagenomic complexity and environmental complexity. Microbes will change in time and space, for sure. The environment will also change in time and space. And if we are unable to collect enough information to account for all this variation in our models, the predictive capacity of any kind of model we create to try to understand what's going on in the system is going to be quite low. If we go a bit narrowing down all these axis of complexity, we have an example of human cohorts, for instance. So if we narrow it down to a specific cohort in a specific populations in a specific species, then the genomic complexity is narrowed down considerably. But still, we have a huge metagenomic complexity and huge environmental complexity. If we go to human twins, for instance, then the genomic complexity has been narrowed down almost completely. We have two individuals that are genetically either identical or very, very similar. So the genomic complexity is very low. So we will be able to start answering some other questions that we were unable to answer when analyzing wild species in a wild environment, etc., etc. But still, humans have free will, and it is very difficult to tell a human what to do in an experiment. So there will be a lot of things that we will be missing as well. You can ask some human patients and say, okay, take this treatment or do not take this treatment, but the rest of the variables that are affecting the biology of those animals is not controllable. So still we have a huge environmental complexity, and the metagenomic complexity can be also very large. If we go down to production animals, let's go to an aquaculture system, for instance, then everything starts becoming more and more less complex, because usually production animals are genetically quite similar, and the environmental complexity tends to be considerably lower as well. If, of course, it will depend if we are talking about salmon in a huge pen in the sea, or juvenile salmon in a tank inside an aquaculture facility, the environmental complexity will be quite different, because in one case we will be able to control the salinity, the water temperature, etc., while in the sea we cannot do that. So there might be considerable differences. And the same happens with the metagenomic complexity. We have some animals that are in a closed environment. The exposure they have to environmental microbes is far smaller than whatever occurs in nature. So production animals usually will be quite low in this complexity gradient, but still, depending on how the production system looks like, it can be quite different. And then perhaps in the very bottom, we have the laboratory models. We have, let's imagine, inbred animals that are genetically identical, could be also germ-free, or we can include whatever microbe we want so that we have full control of the microbiome. And so we can make sure that the metagenomic complexity is low. And as we are in the lab, we can control the photoperiod, we can control the humidity, the temperature, the diets, we can control everything, basically. So the complexity of the system is much smaller. In all these gradients, we need to consider that perhaps when we work with these lab animals, whatever result that we might get can be very specific. We might be able to go to very high resolution questions, but we need to always ask ourselves, is this actually reflecting something that is happening in nature? Is whatever analysis we are doing here representative of the actual question we want to answer? And these are the two edges that we have, from wild species to lab animals. In one case, we have a huge variability that does not enable us to answer very specific questions or some of the very nitty-gritty details, while in the other case, in the other end of the spectrum, we have the opposite. We might be able to answer some very specific questions, but we are lacking the context, and we don't know whether what we get might be extrapolable to many other environments. So let's now zoom in to each of these axes of complexity. So if we focus on the genomes, something we need to consider, for instance, is whether the genome of the animal is more or less complex. Because, for instance, humans, we have a complex genome compared to C. elegans, for instance. But our genome is not complex at all if we compare it to newt, for instance, or even wheat, which are much larger genomes that are much, much more complex. And even for some systems like salmon that has a medium-sized genome, still the genome structure is so complex that it is very, very difficult to analyze. Because here we need to consider not just how large a genome is, but also how many repetitive fragments there are, how many insertions, deletions, how many duplications might have happened, etc., etc. So in some cases, we will have very large, complex, and even polyploid genomes, like in the case of wheat, which is not just large and complex, but also we have 6 copies of the genome in each cell of wheat, which makes everything quite complex. Then we have some cases, like in salamanders and other newts, that genome sizes that are 20 times larger than ours, than humans. So they are really large and very complex. And as I mentioned, we have salmon, for instance, that again, the genome size is similar to ours, but it is much more difficult to work with because this is much more complex than the human genome in terms of how it is structured. And then in the other end, we have C. elegans, and it's not surprising that C. elegans is used as a model species because its genome is small and simple. So we can answer a lot of questions that we would not be able to answer working with salmon or trout or working with salamanders, for instance. And this is the intrinsic complexity of the genomes of the different animals. But we have another level of complexity that we might also consider, which is the comparison that we want to do. It's not the same if we are comparing species and genomic information of 2 different species, 2 different populations within the same species, outbred models that have the diversity, genomic diversity that could be similar to what we would observe in nature, compared to inbred models. So inbred models is, in the end, the animals get crossed with each other so that almost all the variants that we have, almost all the alleles of the genes are homozygous. So there is no variability, and the animals that we use for working are almost identical. They are not clones, but are almost identical. And then we have actual clones in the very end. So we have, for instance, Daphnias that they can generate clones of themselves, can enable us to address some questions where we have full control of the genomic complexity. So this is going to be important for you to consider when you are thinking about your project, your grant proposal, what kind of system you are going to use. You can check how complex the genome is. You can also consider whether it is possible to work with clones, to work with inbred models. Are you going to use some kind of farm animals? What is the genomic complexity that you might have there? So these are things that you need to keep in mind when selecting the system. And if the system is fixed and you always work on salmon and there is nothing else that you do, at least consider what kind of questions you can answer with salmon. If we jump to the next axis, the metagenomic complexity, we have something similar. We have, on the one hand, the intrinsic complexity. So as we mentioned before, there are some systems that have quite complex microbiomes. So if we go to birds, if we go to terrestrial mammals, we are talking about from dozens to hundreds to even a few thousands of bacteria, different bacteria that can be associated with each single host. But there are some other animals. Here we have the example of the bee that have about 10 to 20 different bacteria associated with it. But something similar happens also with salmon or trout. So salmonids usually have a very low diversity in their intestine, even though it can change considerably. And then we have in the other end of the spectrum, we have synthetic communities. So if we are working with a germ-free animal, so let's imagine that you have a zebrafish that you take to the lab and you raise it germ-free, then you might be able to provide specific strains of a specific genes or bacteria that you want to be included in the microbiome. So this synthetic community will have a very low metagenomic complexity. The same way we did with the genomes, we can also divide the complexity of the microbial metagenomes in 2 angles, or 2 axes. So one is the intrinsic one. I would say, okay, how complex is the system within each individual that we want to analyze? And the other one is what kind of comparison we want to do. Because perhaps we are interested in checking the different compositions, which bacteria are different, but also how their abundances change. and how their expression level of the different genes change. So depending on what is the level of analysis we want to do, the complexity of the microbiome can be larger or smaller. If we are only considering whether a taxon is present or not, is Escherichia coli present in A and not in B? That's not a complex question. But if we say, is the expression of carbohydrate active enzymes larger in A than in B, then we need to consider much larger complexity. So depending on how we address those questions, then we need to consider how feasible it is, depending on what is the intrinsic complexity, and then the comparisons we want to do. And finally, the third axis is the environmental complexity. And here we can also split into, one is the intrinsic variation that we have. And as I mentioned before, if we go to nature, if we go to the Mediterranean Sea, you might not be able to control how much the temperature will rise the next summer. We don't really know. Probably it's going to be warmer than this summer and the previous summer and the previous, but there are many other things that we are unable to control. But if we take just a portion of it and we close the system, then perhaps we might be able to control some of the environment. Perhaps we cannot really control the temperature, but if we set a filter in a pen, then we might control for the input and output of some kind of zooplankton or phytoplankton that might be affecting whatever diet of some fish that we might be raising in a pen, in a sea pen, for instance. Then if we go to the lab, we might be able to control much more. And if we have an open environment in the lab, we might be able to control the water supply and the food and the temperature. But still, the animals get exposed to environmental microbes. And we can even limit that if we have a full controlled lab environment in which the animals are raised in a sterile setup, and the entire contact with outer microbes is completely disrupted. So depending on which part of this spectrum of complexity we are, we might be able to answer some questions or others. And again, we have the possibility also to explain or to define the complexity in terms of comparisons. So for instance, we might not have any kind of control of the environmental factors that we want to compare. Let's say we want to say, okay, I want to see what is the difference between summer and winter. So summer and winter are two categories, but are quite vague in the sense because one summer from 1 year can be quite different to another one. So the level of resolution we will have will be quite limited. It can be the same with the variation of multiple environmental factors. We might have an experiment in which we change the temperature, we change the diet, and we change the exposure to iron, for instance. But it is far more defined than winter or summer. But still, we have many variables that are affecting together to the system. And sometimes this is important or this is relevant because if we want to mimic what is happening in nature, changing a single variable sometimes might not be realistic. So because that will never happen or almost never. So for instance, if we are working with comparing temperature, but temperature is always linked with humidity in terrestrial systems, then perhaps it is more explainable. Makes more sense to change temperature and humidity together because this is what happens in nature. And if you are trying to understand a process that happens in nature, then perhaps you want to put both together. But if you want to actually say, okay, I want only to see what is the effect of temperature, then you might be able to control a single variable. And then you have another experiment that you run only with humidity. So depending on what question you want to answer, then you need to consider, am I going to compare season, winter versus summer? Or am I going to compare multiple variables versus multiple variables? Or am I going to narrow it down to a specific variable that I want? And the questions I want to answer is going to be specific to that variable. Then the interpretation of the result will also need to consider this to see whether you can actually extrapolate what you got to any kind of larger question that you were willing to answer or wanting to answer. So now I have 3 questions for you. and I will ask you to join slide again. And there are 4 options here that I would like to ask you to rank these systems from maximum complexity to minimum complexity. So we will have 3 questions. The first one is depending on genomic complexity. Then the next one will be on metagenomic or microbial complexity. And the final one will be on environmental complexity. So the first scenario is a clonal population of corals that are exposed to 2 different temperatures. So we have corals in the lab, and we are exposing them to these 2 different temperatures. Then we have another system that's it is 2 tuna species. One is from the Mediterranean and the other one is from the Atlantic, and we are trying to understand what are the differences between this. We also have 2 tanks of commercial rainbow trouts that are under different dietary regimes in a commercial environment, so aquaculture environment. And finally, we have a clonal population, wild manatees that are captured in a single mangrove. So according to genomic complexity, how would you rank them? 1 is maximum, 4 is minimum complexity. All right, so no one is ranking anymore. So what you selected is that according to genomic complexity, the most complex one will be a species of wild manatees captured in the mangrove. Then the next one, 2 tuna species. Then the third one would be the commercial rainbow trout. And the final one is the clonal population. So from my point of view, so I would agree with you that the clonal population is the one that has the lowest genomic complexity, because we are talking about individuals that are genetically identical. And I would also agree with you that the third one should be the 2 tanks of the commercial rainbow trout, because usually when we work with aquaculture species, the animals, even though they are not genetically identical, they are genetically very, very similar. For the options 1 and 2, I would disagree with you. And I would disagree because in the first case, you say that the wild manatees, we are talking about a single species in a single mangrove. So even though it is more complex than a commercial setup of rainbow trouts, the genetic complexity that we have on manatees, it would be complex, but not as complex as comparing 2 different species from 2 different environments, like the tuna species. Of course, we don't have the information of how complex the genome of the manatee is and how complex the genome of the 2 tuna species is. So it might be that the 2 tuna species genomes are very, very simple, and then we can say, okay, the manatees are more complex. But if we just simply rank it by comparing 2 different species or a single species in a single population, then I would say that the 2 tuna would be more complex than the manatee. So next question is, we have the exact same 4 systems, but this time I will ask you to rank them according to their metagenomic complexity or microbial complexity. First is the most complex microbiome. The last one is the least complex. And use different criteria. I know that you don't know the microorganisms, that the communities that they have, but based on whatever you can think about, how would you rank them? Okay, so also in this case, you decided that the clonal population of corals is probably the one with the lowest metagenomic complexity, and I would agree with that with you, because usually corals associate with one or a few dinoflagellates, and they don't have a huge microbial complexity. And if we have them in the lab and we are experimenting with them, most probably the complexity of the microorganisms associated with them won't be that large. With the other three, I do completely guess that with the information that we have, it is very reasonable to rank it as you have it done, because we have two tuna species in the environments in the wild that most probably they have a lot of different microbes, etc. While in the tanks of the commercial rainbow trout setup, the exposure to microbes is far smaller. But there's one thing that I didn't mention before, and this is something to also consider in this, that manatees are herbivores. So when we are usually talking about herbivores and carnivores like tuna or rainbow trouts, the complexity of the microorganism communities of herbivores tend to be considerably larger, like considerably larger. So both tuna and rainbow trouts, they have a rather low complexity microbiome. Usually, they might be different across different species, etc. But usually carnivores, both in the sea and also in the terrestrial environment, have lower complexity of microbiome. While herbivores tend to show considerably more complex microbiomes. And the reason is that usually animals do not have the intrinsic capacity for degrading complex polysaccharides derived from plants. And the only way to digest those compounds properly is to have microbes that will do that work for the hosts. So while recognizing that without that information, the way you ranked this would make perfect sense, probably perhaps shifting to 2 and 3, considering that the manatees are herbivores and the tuna and the rainbow trouts are carnivores, perhaps we should rank the manatees first, then the 2 tuna, then the 2 rainbow trouts, and final clonal population. And now the third question is, again with the 4 systems, but ranking them according to their environmental complexity. So in this third case, we are losing participants as we go ahead. So I hope you're all doing fine. So there is no much controversy here. I was expecting to have a little bit more of a controversy between 3 and 4, but looks like most of you considered that the complexity that different temperatures entail is larger than what dietary changes entail. And I guess this might be coming from considering that diet might not be an environmental factor. I would argue that in this context it is. And when we are talking about microbiomes, actually diet is one of the most important environmental factors, because depending on which type of dietary elements, what kind of compounds you insert inside the animal, the microbiome can change like really significantly. So in this case, I would personally rank the dietary regimes over the 2 different temperatures. Although, if the temperature differences are really large, then we might be start observing bleaching of the corals. And this is perhaps what you had in mind. Say, okay, if the temperatures are too high and then the acidity of the water changes, and then perhaps the corals are unable to actually recruit some bacteria with them or some algae. So it can be, depending on what we are talking, it could be one or the other. So that's why I was expecting a little bit more of controversy, but perhaps you are more sensitive to corals than to rainbow trouts, and that was a reason. But I would agree with you that the other 2, in one case for the wild manatees, we have a mangrove, a single one that will have some spatial variation depending on where the manatee lives, and there will be also a temporal variation across the year, and depending on when we sample them, the environmental complexity will be larger or lower. And in the first case with the 2 tunas, we have in the end 2 different seas that we should expect different temperatures, different acidity, different exposure or different access to diet, etc., etc. So there would be no much controversy in that case. But well, these were the 3 axes that I wanted you to consider for your projects, and depending on what kind of questions you want to answer, please keep this in mind. Do you want to go broad but with very minimal control over what you are experimenting with, or you want to go to very specific question, then you need to control as much as possible your system. Now the next session is on a slightly different topic. So if you have any questions for this part, please raise your hands before we continue.
Still no questions.
No questions.
No.
Okay, so we will continue then. So I have included here a few examples of different approaches that we use in our group for doing research, but could be also relevant for you for considering what kind of project you would like to consider for your assignment. So one approach we can use is just field observation. So we might want to know what is out there. And oftentimes this is the first step. If we don't really know how nature is structured, it is very difficult to have super detailed hypotheses about what is happening. So this might be the first step in the way to understand some process. And when we say a field observation, in our case, we have this initiative that is called the Earth Ologenome Initiative. And I think it's a good example for seeing what kind of questions can be answered and what kind of work can be done based on field observations. That also give us some important considerations of what are the limitations of this type of approaches. So what we do in the Earth Ologenome Initiative, and this is mainly coming from the realization that there was a very fundamental problem in the microbiome, in the wild animal microbiome community a few years ago. A lot of data were being generated, but these data were barely comparable. And the problem is, as we will see in a few minutes, that mainly when we are doing microbiome research, the sampling, the sample preservation, the sample processing methods can introduce huge biases. So we really need to standardize the methodology to analyze something that actually makes sense. And this was one of the main considerations for creating the Earth Ologenome initiative. This had been done before within the Human Microbiome Project, for instance, or even the Earth Microbiome Project. But associated with wild animals, there was nothing similar. So this is why we created this Earth Ologenome initiative with one very particular distinction, which was that every sample that all the people participating in the Earth Ologenome initiative have been taken with the exact same procedure, stored in the exact same buffer, and processed in the exact same way. And all the metadata that have been collected also have been standard, so that we can now create a huge data set of comparable data that enable us to do this type of analysis we want to do from field observation data that it was impossible to do beforehand. So our database right now looks like this. Each dot is a different sampling point, and the colors indicate what species has been collected. You can see that we have a huge bias towards Europe. And the reason is that something that you will need to keep in mind in the future. Nowadays, the transfer of genetic resources, and genetic resources is any kind of biological entity that contains genetic information, it is very much regulated and in some cases restricted. So within Europe, it is quite easy to work, but once we get out from Europe, there are many countries that have different type of access and benefit sharing rules that make quite complicated to get samples out from there. So for instance, it is very difficult to get samples out of Turkey or from India, China, in some cases also Brazil. So this is something that also needs to be considered when setting up a project. So if there is something that can be done in Italy, don't go to Turkey. It's going to be much simpler for you to get the data in your own country, because otherwise you will need to enter into a lot of paperwork that we have to do it, because as we are working at a global scale, we need to go through this. But I'm telling you from experience, it can be very complex, because each and every country have developed their own rules. In many cases, this stems from the Nagoya Protocol, and many countries have signed the Nagoya Protocol, and then they have created their own rules, national rules, based on the Nagoya Protocol. There are some other countries that do not adhere to Nagoya Protocol, but have their own rules. And then there are some other countries that have nothing, like the United States, for instance. They have their own, the different states might have different rules, but there is no one ABS regulation that as such. But this is something, just a parenthesis, that if you can work or you can have a question that can be answered by a system that entails less paperwork, try to do that. There are some questions, though, that we cannot really answer by just observing what is happening in the nature. As we mentioned before, when we go to the wild, there are so many environmental variables that we do not control. There is so much genetic variability that we don't know about. It is very difficult to run some studies and answer some questions that we have in mind. Sometimes we need to work in captivity and run experimental work with them, because while we might see correlations between any kind of genomic or metagenomic and environmental factors, setting that one is the cause and the other one is the effect is almost impossible. We might see, and we often see, changes in the microbiome composition, in the microbiome diversity, in the microbiome functional aspects between two populations, but whether these changes convey any kind of biologically relevant effect on the host cannot be known. And the only way to do that is to take these animals to the captivity and, for instance, conduct fecal microbiota transplants. So we can take the microbiota of some set of animals and transplant it to another set of animals. And this is what we did in some of these experiments that I will show you in the next slide, I think. The important or the most relevant aspect of this is that once we start experimenting in a much more controlled environment, we can start addressing much more complex questions. In this case, in this experiment that we ran 4 years ago, we analyzed the microbiome dynamics of two species under exact same conditions. So we had Crocidura rusula, which is a shrew, and Apodemus sylvaticus, which is a mouse. And what we saw is that the functional attributes of the microbiome when these animals are exposed to an identical concatenation of environmental stressors can change quite differently. So this is very difficult to do in the environment because we don't really know whether each animal we have sampled at each moment has been exposed to the same environmental conditions. Perhaps Crocidura rusula has been under the ground and Apodemus sylvaticus has been in a tree. And then the temperature conditions might be completely different. They might have been exposed to different type of pathogens, etc., etc. If we take them to captivity, then we might run experiments in a much more controlled environment. And as I was mentioning before, we can also do this kind of fecal microbiota transplants, which in the case of animals, we can do that by gavaging. In the case of gavaging means that we can prepare a mixture of microorganisms derived from feces from some animals, and then we can actually force the animals to ingest that. And this is something that it is done very commonly in mice, but we have also done it with amphibians and reptiles. If you are working with aquatic animals, usually what you can do is to enrich the water of that tank with bacteria that are coming from some donor population. So you try to clean up the environment first, and this is usually done by adding antibiotics. And once you have reduced the load of bacteria there, then you would enrich that tank with water contaminated with bacteria that are coming from other species. The smaller the tank is, the higher will be the chances that your animals, that could be zebrafish, or could be some kind of corals, or could be jellyfish, that will be associated with this bacteria. But this can only be done in this kind of setups that are close and can be controlled very clearly. And then we have a third option that is more advanced, but is to try do this kind of work in vitro. And even though this has been almost impossible until a few years ago, nowadays there are some systems that enable us to somewhat approximate to having in vitro systems, so non-real systems that emulate real systems. And in this case, in our lab, we work with gut-on-a-chip models that we create from different animals, and then we can add different bacteria, or we can add different MAMPs. You might remember that from the first session, these microbe-associated molecular patterns that enable us to see how the tissue from different animals react to the exact same stimuli. So this enabled us to answer much more refined questions, because we can see, okay, if we have a rainbow trout and we have a salmon, and we get intestinal stem cells from them, we create synthetic gut-on-a-chip models that are coming from organoids, and then we can expose them to the exact same compound that is coming from a specific bacteria. Let's imagine that we are talking about mycoplasma, and we want to see whether the mycotoxin of mycoplasma is triggering the same type of reaction in the rainbow trout and in salmon. So then we can create these gut-and-chip models, and we can experiment with that in the lab. And this is something that we are doing with terrestrial animals, but in the future could be potentially done also with the aquatic animals. I won't get too much in details on this because it is quite complex in that sense. The next part is about the next phase. Once we know what kind of experiment or what kind of study system we want to and study approach we want to use, are we going to do experimental work? Are we going to do field observation? Are we going to do in vitro experimentation? There are a lot of other considerations that need to be taken into account to generate reliable data. And this mainly relates to microbial metagenomic data, because the host information, of course, if we are talking about gene expression, it is slightly different. But if we are only interested in the genomic composition of hosts, that's not that variable, and that it's considerably more robust, and it's not that problematic. But when we are working with microbes, generating reliable data, it's quite tricky. And we will go through this in the following minutes. So 2 years ago, we published this paper in which we presented this extremely complex figure as the first figure. What we wanted to convey with this figure is that whatever you do in the field, they have the field here, or in the laboratory, have huge impact on whatever you will find out when you do the bioinformatics and when you do the data analysis and the statistics. So we try to set the dots and link the dots between what happens in the field and what happens in the laboratory and what happens in the final results, so that people could be aware of any kind of biases, limitations, filterings that might be distorting the results and might lead us to make the incorrect interpretation of the result that we have. So what we did in this publication was to list 15 potential pitfalls that related to 3 different domains. The first domain was related to the intrinsic properties of the samples. The second one is whatever we do in the field and the laboratory. And then the final one is related to the study design, how we structure our study, and how can the study design lead to different kind of biases when we generate the data. So we will go through these 15 points step by step. And the first 5 points are linked to the intrinsic properties of the samples. And we tried to abridge all this information in this huge figure that we will go through step by step. Because the first thing we need to take into account is that the microbial community that we are trying to characterize might be altered already before sampling. So one thing that perhaps we need to consider is that, are we actually sampling the community itself, or are we sampling a proxy for that community? We might be interested in the gut microbiome, but what we might be able to get is fecal samples. Let's imagine that we are trying to analyze the microbiome of cetacean or of a seal. We might not be able to kill those animals or to sample the intestine of those animals. And the only thing we might be able to do is to get the fecal material and analyze the fecal material as a proxy for the intestinal microbiome. So we will need to assess whether the fecal material is a good proxy for the intestinal microbiome, because we might be already distorted, and whatever we are interpreting as intestinal microbiome, it's actually just the fecal microbiome. And there might be significant differences between both. Another source of distortion is that whether the sample we obtained is fresh or not. Sometimes if we are working in the lab, we might be able to collect the samples as soon as they are generated. But sometimes we might be perhaps in the field and we might be collecting the fecal material minutes or hours after it has been produced. And this time can entail big changes in the microbiome composition of that proxy sample, because saprophytic bacteria will grow, while some other bacteria that were, for instance, relying on low oxygen levels might become shrunk because they are exposed to oxygen and it's not the same environment anymore. We might also be interested in the temporal dynamics, that perhaps there are some seasonal or daily dynamics that might differ the microbiome. And we might be interested in what is happening when the animal is active. But if we sample when the animal is inactive, because it is more appropriate to do it this way, then the microbiome might be different. So we need to consider whether these kind of differences are affecting the results that we want to get. And one final important aspect is that the microbiome itself might have some spatial variation. So if we collect, for instance, a fecal sample that whale have generated, and we find it floating on the sea, most probably the outer environment of that material will be more contaminated with environmental bacteria than the inner part. That perhaps it's more reliable to characterize the intestinal microbiome of this bacteria of this animal. So these are some of the aspects that you would need to consider when you design a sampling scheme to ensure that all these aspects are not affecting the results, or at least you can control for them, and you can account for them in the analysis that you do. Another potential problem we might have is that the system we are analyzing have very low levels of DNA, of microbial DNA. Let's imagine. So sometimes we have some environments that we sample that have really high biomass, really high diversity. But we have some others that have very low biomass and very low diversity. When we do the laboratory processing, if the lab processing is not good, then we might be losing a lot of the diversity. And this is shown in this example here. Let's imagine that the first one is we have, it can be, let's say, the intestine of the rainbow trout that we are sampling. And we might be expecting some diversity here. And then we have some juvenile rainbow trouts that have less diversity. But in this case, the laboratory processing has been suboptimal. The DNA extraction or the library preparation has not gone as we wished. And the result of that inefficient laboratory processing is that we actually lose a lot of the DNA that was there in the first place. So we might end up having a system that looks very much to this other one that was originally low biomass and diversity, even though the original sample was high biomass and high diversity. So this kind of suboptimal laboratory processing can actually impact the final results. And if we are unable to really pinpoint what happened in the laboratory, then we might be misinterpreting the results. And this is becoming more and more likely to happen because very few people tend to process their own samples for DNA analysis. So what we usually do, not in our case, because we are very picky on this and we have the facilities for that, and we have our own facilities for doing all the DNA extractions and library preparation and the sequencing, etc. But most people don't have access to that. So they need to rely on external companies or collaborators that do this. Oftentimes, you don't really know what happened in the lab. You don't really know whether the DNA amount that you got in the sample was low because the extraction went wrong or because it was low in the initial sample. And without that information, then it is very likely that you will misinterpret your results. So this is something to keep in mind. Sometimes we don't have any other option, but the more information we request to the companies or to the collaborators that are generating the data, then it might help us to interpret the results. And for us, a very clear example is that whenever we see an outlier, we always check whether that outlier has some kind of funky laboratory metrics. So perhaps the DNA amount that was extracted was as good as the other ones, but then after the library preparation, suddenly the molarity that we got for sequencing was far lower. So most probably that entails that the library preparation for sequencing went wrong. So then we know, or we are quite certain that what we observe, that outlier, is due to a technical aspect and not to a biological aspect. If it's a technical reason, then we will discard it. But if it is a biological one, then we might want to cut it because it is actually showing what is the environmental or the natural complexity of the system that we are working on. This is the problem that we have with low DNA, but we might actually face the opposite problem as well. And this is perhaps quite counterintuitive, but having high microbial diversity is sometimes problematic. And it is problematic mainly because if we are trying to reconstruct bacterial genomes for complex metagenomic mixtures, if the complexity of the sample is too high, then the computational methods that we have for doing what is called the metagenomic assembly and then the binning, they just fail. And we might not be recovering the entire diversity that we have there. So it does happen sometimes that even though you might have a system that is hyper complex, then you end up having a representation of that system that is far less complex because the metagenomic assemblies were unable to resolve all this properly, and then you end up discarding a lot of data. So this is something also to consider. Some cases, and it happens sometimes in some water environments, the amount of different microbes that you have is so large that it can become problematic to analyze. Another source of problem is the ratio between the amount of microbial DNA and the amount of non-microbial, or in this case, host DNA. And this is a very huge problem with fish. And if any of you have worked with fish and microbiomes, might have faced this issue, mainly if working with shotgun metagenomics. So if you are trying to sequence the entire amount of DNA you get from the sample, you will end up seeing that most of the data derives from the host animal. And usually this happens when the microbial biomass is very low and the host is releasing epithelial cells. And this happens in a lot of intestinal environments. If we have an animal that does not have huge microbial biomasses, huge microbial densities in the intestine, usually there is far more host DNA, fish DNA in this case, than microbial DNA. And that might entail that if we are working using shotgun metagenomics rather than approaches that are based on sequencing of targeted genes, then the sequencing might be overloaded of host DNA. And then we have very, very limited amount of microbial DNA that enable us, so that prevent us from properly characterizing the microbiome. And this is something extremely common in water environments and with water animals. So when considering a project, for instance, with fish and their gut microbiome, if that's what you will go for, probably you will need to budget for a lot more sequencing than what you would need for a mouse, for instance, or even for a human. In humans, usually about 90–95% of DNA that we get from the fecal samples is bacterial DNA. In salmons, it is less than usually 2%. It's 98% salmon DNA, 2% bacterial DNA. And this is a problem because sequencing is not cheap. It's becoming cheaper and cheaper, but it is not cheap. And if we need to sequence 50 times more to get the same amount of information, then we have a problem. So this is something to consider. And one final consideration regarding the initial intrinsic properties of the samples is degraded DNA. In some cases, the sample, if we are collecting it from the environment, the DNA itself can be damaged. And when the DNA is damaged, then assembling the different reads to reconstruct bacterial genomes is far more complicated. This is mainly relevant for ancient or historical DNA research, not that relevant for marine environments, I would say. But if you are relying on historical collections that are present in a let's say, natural history museum, you are working on microbiomes from the dental plague of seals that were, I don't know, cataloged 100 years ago, then the DNA you might find in the teeth of the seals is, for sure, it's going to be degraded. So that will also entail that the reconstruction of its microbiome will be also inefficient, or at least not optimal. Do you have any questions on these first 5 points? I know that some of these topics might be very technical, and as I don't really know what is your background, so apologies, but feel free to ask any questions if you feel very lost.
No questions.
No questions. Next 5 points, I think. Again, or 6 points? 6 points. And this relates to laboratory steps. So once you collect your samples and you take them to the laboratory, still you might face problems. And this is something to consider. The first problem you might be facing is related to what I was mentioning before with the Earth Ologenomics initiative, and relates to the collection, preservation, and sample preparation biases. So depending on how you collect the sample, what kind of buffer you use, or you just snap freeze it, or you keep it at room temperature, or you might be sampling in the tropics and then the sample is at 40 degrees, or you compare to sampling in the Arctic and the sample is frozen, then the results can be very, very different, because the decay of the quality of the samples can be faster or slower. If you are using a preservation buffer, that decay will be lower. If you are freezing the samples, that decay will be lower. If you don't do anything, the sample probably will undergo a lot of variation compared to what you got on the first place. So it is important to consider what kind of collection method you're going to use, what kind of preservation method you're going to use, and what kind of sample preparation biases you expect. You should expect also biases in the DNA extraction. So when talking about bacteria, we are talking about organisms that have quite different structure of the cell wall. Usually the bacterial cells are considerably more tough than the eukaryotic cells, because they, not all of them, but most of them have cell walls, but they have cell walls of different types. And the different types of cell walls have different kind of robustness or toughness. And depending on what kind of DNA extraction method you use, you might be able to get an overrepresentation of some bacteria with some type of cell wall compared to others. So usually gram-positives are tougher than gram-negatives. And of course, they are tougher that the bacteria do not have a cell wall, like mycobacteria, for instance. But also it relates to the cell form. There might be some bacteria that are in the spore form that are really, really tough, and we might not be able to get any DNA out from there. While if they are in the vegetative state, then it's going to be much easier to get the DNA from there. So this is something to consider, that depending on what kind of methodologies we use, then the extraction can be biased. Usually for bacteria, it is essential to use mechanical lysis. So what we do is a combination of chemical lysis and mechanical lysis. And mechanical lysis entails that you add small bits, could be our silica bits and aluminium bits and different kind of structure and density, and then you shake the sample really, really toughly, really, really intensively. And when the cells get just shocked by these bits, then they tend to lyse and the DNA is released. If this extraction method is too tough, then you might also end up breaking the DNA. And when you break the DNA, your DNA fragments are smaller, and then you might also have some issues related to that. So it is an important point whenever you are doing DNA extraction from microbes to consider what kind of extraction method you are going to use. It is also important to consider that whether the DNA extracts that you get are clean enough. Sometimes you might have inhibitors, so different kind of molecules that are also extracted with the DNA that might be preventing the downstream enzymatic reactions to happen properly. And this might be related to library library, but it's library ligation, or could be also library amplification. If you have an inhibitor that is affecting a DNA ligase or a DNA polymerase, then the downstream library preparation will be affected, and what you will get will not be representing properly what you got in the first place. So the DNA extraction is not only about how much DNA you get, but also how free it is from inhibitors that affect downstream processes. So this is something also to consider. As I mentioned, if we degrade the DNA also too much, then we might have extremely short DNA fragments, and having short fragments is not good for metagenomic assemblies that we will see later on. If we have very short fragments, it's much more difficult for metagenomic assemblers to create longer sequences, and that way the quality of the genomes of bacteria that we reconstruct will be considerably smaller, or considerably worse. There are many other aspects that might be affecting the library preparation. This is probably too complex and I will step over that. But there are also aspects that are related to the sequencing. So depending on what type of sequencing method we use, we might be able to recover better or lower quality genomes. So it is important to also consider, am I gonna use just short read sequencing? Or am I gonna use PacBio or Oxford Nanopore long read sequencing? Or am I gonna use hybrid sequencing? So these are other considerations that you might also need to make when considering what type of DNA sequencing you are gonna do for your project. And then we have the final part, which is related to the study design. And in here we have 4 different considerations. So the first one is whether we have environmental and reagent contamination. And this is a huge, huge thing in microbiome research, because microbes are everywhere. And mainly if we have samples with very low biomasses, like what I was mentioning with salmon or the rainbow trout, but the same happens or something similar happens with clams or even jellyfish, the amount of bacterial biomass that we have is usually quite low. So it is very easy to get it confounded with environmental contamination that could be present in reagents. A lot of the reagents that we get, if they are not ultra clean, contain bacteria at very low densities, but contain bacteria. And this might be actually affecting the representation that we are doing from our samples. Of course, if you manipulate the samples with your own bare hands, your hands also contain a lot of bacteria. So you will be transferring those bacteria to the sample, and then you will be representing also part of your skin bacteria in your sample. And this is something you don't want to do. So when working with environmental samples for microbiome research, always wear gloves, ideally masks, and ensure that you are using very clean reagents, because otherwise you won't be able to get reliable results. And this mainly affects when you are working with systems that have very low bacterial biomasses. If it is a lot of the bacterial DNA, probably the effect of the environmental contamination will be low. But if you have very low amount of DNA in the first place, then the effect of the environmental and reagent contamination will be much larger. Another thing to consider is cross-contamination. Usually when we work in the lab, we don't work one by one with each sample. You don't work in a closed environment that you first go for the first sample, and then you go for the second sample. Instead, you work in batches. And depending on how these batches are used and how they are processed, the likelihood for cross-contaminating these samples might be larger or lower. You might consider that 2 samples that are located in 2 wells in the same plate are much more likely to share some contamination with each other than compared to another one that is farther away. And sometimes it is not just, so you don't really need to drop a liquid in the incorrect well to introduce contamination. It's enough for having some sprays. So for instance, when you get the pipettes out and if the drop comes down to the well, but then creates some kind of splash, then it is very likely that you will end up contaminating the sample. So this is something to consider. When we work with robots, We do have cross-contamination, but it's a cross-contamination that can be modeled because it is exactly the same all the time. So we might expect that the contamination will happen at this proportion from S1 to S2, from S2 to S3, etc. in this sample. When we are working manually, that is much more difficult to control because no one works exactly the same way all the time. So these are the pros and cons that we have with in terms of cross-contamination. Another important thing to consider are batch effects. So we know that depending on what lab reagents you are using, depending on who is the operator that is actually doing the work, the results look different. And it's a pity, but it is that sensitive oftentimes. So it is important to consider how you are going to randomize the samples to ensure that the batch effects are not confounded with your main variable of interest or your main question of interest. If you are contrasting population A to population B, don't run all the samples from population A together and all the population B together, because then the technical bias, the technical batch effect will be confounded with your variable of interest, with your comparison of interest, and you might not be able to tease apart whether what you are seeing is a technical bias or an actual biological difference. So this is something to consider. And the last one is the presence or the absence of biological replicates. So all these steps, all these laboratory steps can introduce biases, as you might have already, as I might have already be able to convince you that the lab steps are very critical. If you don't have biological replicates, what you might be getting is a distorted representation of your sample that you don't really know how distorted it is. If you have biological replicates and you do the same analysis in different batches, then you might be able to see whether what you get is reliable or not. Because if you do something 3 times and you get the same result, then you are really certain that what you get is correct. If you do it two times, well, you might be quite certain. If you do it just once, you don't really know. So this is something that it's also considered. It needs to be considered. It is true that introducing biological replicates entails spending more money, spending more time. So then you need to assess whether introducing biological replicates is essential, whether it is important, whether you have budget, whether you should be investing in this compared to other things. But this depends on what is the study question and the study system. So that was it for this session. It was a little longer than the previous one, but this is the longest one. So then we should be able to take it to the schedule. So what I would like to ask you now is to think of this question and think of which of these problems that we mentioned, these 15 questions, you anticipate you might face using your samples. So if you have already started with your project or you are expecting to have a project, yes, even though you might not be working with microbiomes, try to make this effort and say, okay, if I am working with this jellyfish, if I was to work with microbiomes, which of these problems would you anticipate you would have? So I believe this is an open answer poll. So I will give you 3-4 minutes to think a little bit, and please add your response here. And then we will just see what kind of systems you are working with or you are expecting to work with, and we can have a chat on this. And in the meanwhile, I will go to the toilet and come back in a second. So I can see that a lot of you are concerned about contamination. You can see there are 14 people so far have mentioned contaminants, and it's a huge source of a problem, but luckily is one that usually can be minimized a lot. So just following some good steps, then it can be minimized. And if you run also some preliminary analysis to assess what is the effect that contaminants might have into your results, then you might be able to adjust the measures you want to use to the problem, because sometimes we might be overstating the problem, and but usually when working with microbes, it is an issue. So it is important to keep this in mind. Then I guess this sample mainly refers to sample preservation. And this is important. Or if you are going to be the ones collecting the samples, make sure that you have the correct way already set. And if you will rely on multiple collaborators to get into samples, ensure that they all have the same guidelines, and they all, ideally, that you prepare all the tubes. And this is what we do. And this is, I think, a very good advice, that because it's not only about how well the sample is preserved, but if you also prepare the samples with your own codes, etc., then it is much easier to make sense to the data you get. Because if you're expecting to collect hundreds of samples from different collaborators, and each uses different coding systems, etc., it can easily become a mess. And then the result for the decisions to be made, referring to cross-contamination, is usually pay attention on the batch effects. It is very difficult to minimize cross-contamination. Of course, there are some general rules, like whenever you are working manually, keep the tubes that you have closed as much as you can. Only open them when you are going to transfer the DNA or the sample. When you are also doing sampling in the field, don't leave the tubes open for minutes, because you are going to get the sample contaminated. But the important thing, I would say the most important thing, is always randomize the samples as much as you can, and ensure that the batches that you are processing are not exactly overlapping what the main question that you are trying to answer. Because it is very likely that a huge part of the effect that you're observing is coming from technical I would say, ways, rather than the actual biological one. Cool. So we are done with this second session, and I would like to ask you one final question, which is the same as in the first session, and is how much of the explanations of these experimental designs for ologenomics did you understand? Whoa, that's a good one. Be honest, please. The best thing of this is that almost everything that I explained is captured in one paper that I can share with you. So you will be able to go through all these points slowly. And if you have any questions, you are very welcome to ask. Like I see, the overall score is very similar to the first one, 7.4. If you have any questions, now is the moment to ask. Looks like no again. So shall we now have a break until noon and restart with the next session at 12? And I think we will be able to do the next one all together. So from noon to 1:30 without a break.
Fine, thank you.
So I'm ready whenever you are. Just let me know.
Okay, we can continue.
Excellent. I hope you see the slide, right? I need to.
Yes.
Yes. Super. So now we will jump from study design and laboratory methods to bioinformatics methods. So perhaps for some of you, this will become more complex. For some others, might be easier. So hopefully we manage to keep that understanding level at 7.5-ish. But I can anticipate that if some of you have not been exposed to bioinformatics so far, yeah, it can be a little bit tougher than previously. But let's give it a go. So I mentioned that the previous session was mainly focused or inspired and based on a paper that we published a couple of years ago. This other one will be based on this other one that we published the last year. And the reason is that, well, I always try to condense information that I consider relevant for lectures in papers, and then it is easier to share all this knowledge with other people. So this one was published the last year, and you will get access to it as well. So if you miss something, then you can go and read this paper. Although I might say that this is the paper will be more complex than what we will consider now in this lecture, because there is no time for everything. But to show the degree of complexity, this is figure 1 for this paper, and this maze actually makes sense, even though it might feel like it does not. And we will try to navigate through 2 aspects of this maze. And the first one is the host aspect that you can see here in the host only part. And then the second one we will go through is the microbiota only part. So we won't get too much into the complex interaction of both, because yeah, things might get too complex. But the important thing is that depending on what type of information you expect to samples, then you will need to make some decisions, and we will go through those decisions. starting from the host genomes. So when we work with samples, we often, if we want to reconstruct some kind of information of the genomic structure of a species, then we usually aim for samples that only contain host genomic information. And this is not only, but at least primarily contain host genomic information. And this is often blood or could be tissue. So if we go for fecal samples or intestinal samples, usually we will get also a considerable amount of bacterial DNA, of non-host DNA. That would complicate analysis. Sometimes this is a problem, sometimes it's not that much of a problem. But if we want to do population genomics, then it is best that we either rely on blood or on the tissue samples, where the presence of microbes should be minimal. And then the first question that we need to ask ourselves is whether there is a reference genome available. And we'll go through that. So what does a reference genome mean? So if we go to NCBI genome, and I will ask you to do that soon, then you will be able to search for different taxa and see whether there is genomic information available for them. So here, what's we see in the circles is this is actually the amount of genomic information, or different amounts of genomic data sets that are present in the NCBI genome. So 2.86 million genomes are included there, out of which almost 40,000 are reference genomes of different species. And we will go through that in a second. So whenever we are generating a genome from a biological sample, we do metagenomic, we do genomic assembly. So we try to get sequencing reads attached to each other to create long strings of DNA sequences. So depending on how well we achieve to create long sequences that are long enough to represent the bacteria, the animal genomes in this case, or the plant genomes, then we can classify them in different categories. So the quantic level is the lowest level of host genome quality. If you find in the reference database that there might be a genome, but it's on the quantic level, it means that usually it's going to be just short read sequencing and no other type of data for putting together how the short sequences next to each other. So this might be good enough, for instance, if you want to get rid of the host DNA and only keep microbial DNA if you are starting from a mixture of host and microbial DNA. But if you want to do some kind of population genetic analysis, if you want to get a population structure, heterozygosity levels, FST values, etc., then quantic level genomes are too low quality, and you will end up having very little amount of information. The next level is a scaffold level, and scaffold level means that quantics of different length are being linked to each other, so we create a little bit better quality genomes, but it's still quite limited. When we are talking about a reference genome, which is representing the genome of a species, usually we aim for chromosome level or complete bacterial reference genomes. Usually most of the genomes we have available nowadays are chromosome level genomes. It means that the genomic sequences have been resolved at the chromosome level, or almost at the chromosome level. There might be some variation, some parts of the genomes might not be resolved. So for instance, the major histocompatibility complex, which is a hypervariable environment in many species, is extremely difficult to sample because there's so much variation, so many repetitions, etc., that part perhaps is not resolved. But we know where this part is. We know in which chromosome it is, and we know where it is. So we don't have a complete genome because there's some variability, but at least we have the chromosome structures properly defined, and we know where the well-known genome chunks are and where the problems are. If we manage to resolve that, then we get a complete genome where the entire genomic content of that the species is known. So these are the 4 levels that I believe that if has not been changed in the last moment, that NCBI also uses for cataloging the quality of the eukaryotic genomes. And what I would like to ask you now is to go to NCBI genome and search for either your study species that you are using your PhD, or you are planning to use in your PhD, or some kind of a species that you like it. And then add here what is the name of the species and what is the quality of the genome of what is the quality of the best quality genome that you find for that species. If you don't find any, add the species and say, okay, there is no reference genome available. If there is a genome that is from the same genus or from the same family, you can also add that information. Say the same species is not available, but there is one that is from the same genus. So if you type in your browser NCBI genome, you will find the link, and there in the browser, in the NCBI genome, you will need to type the name of the species and find out which genomes are available and what is the quality of those genomes. You have 5 minutes for this. If you have any questions, feel free to type them in the chat or raise your hands, and we will try to solve that. Okay, thanks a lot. We got at least 13 answers, 15 species with genomic information. So I think the results are actually reflecting a very common bias that exists with reference genomes. That usually, if we are talking about a species with commercial interest, then a reference genome will be available. If we are talking about a species that is emblematic, the reference genome also will be available. But if we are talking about a species that does not really have a huge societal impact or relevance, the reference genomes are not available. And there are efforts that are undergoing that every year the number of chromosome level genomes is increasing, but still there's a lot of species, the large majority, that are lacking a high quality genome. There were some surprises, like I saw that for me, like I think it was the C-pen that I wouldn't be expecting that it has a reference genome available, but it does it. Something quite common that the Desmophyllum coral doesn't have it, so that's also very common. So the lower we go, also in the organizational complexity level of the species, we have more diversity, but also the representation of the reference genomes is lower. So this is just to tell you that depending on which species you are working with, you might have access to a reference genome, or you might not, which will give you access to a different type of analysis you will be able to do. Because if you have a reference genome available, then you can do genome resequencing, which means that you can just use short read sequencing, and you can map the short reads against that reference genome that exists for your species. So each of these thin lines is our short read sequence. It could be a Illumina sequencing that you map against the reference genome. And when you map there, what you can then do is variant calling. So you can call whether there are differences in the base representation between the reference genome and your sample. And this way you can create this kind of population structure analysis. You can do PCAs to see how the different individuals are differentially structured in a PCA. You can also run different types of statistics of heterozygosity levels, FSTs, etc., etc. But this is something that you can do if you have this reference genome that is usually at least chromosome level. Sometimes with the scaffold level analysis, you can do an approximation to this, but the quality is not the same. So ideally you need to have a reference genome, and many people are making this call for the funding agencies at the government, etc., on investing in generating reference genomes, because it is very important for doing a lot of different analysis. And this mainly refers to species that are not those that have the highest commercial value, but might have a huge environmental value, that we need to learn as much as possible about their genomes, and then use that information to deepen into the population structure, adaptation signatures, etc., etc. One big initiative that is trying to do that is the Earth Biogenome Project, which is an umbrella project that is encompassing multiple reference genome generating projects. It's the Darwin Tree of Life, the B10K, the BAT1K, etc. There are many initiatives all over the world that are trying to generate reference genomes for as many species as possible, but still, most of those are linked to vertebrates, and mostly terrestrial vertebrates. So marine environments are really underrepresented. So it is something to also consider when you do your analysis, that it is likely that your species perhaps might not be already containing a reference genome, but there are possibilities to also ask and contribute with samples to generate these reference genomes. And in the future, we hope that there will be more money that will be put into this effort, because it is really a fundamental information that we need for doing so many analyses. So this is what we have. Once we have a reference host genome generated, then we can map the reads against that genome, and we can generate population structure information. But not only that, we can also search for signatures of adaptation, for instance. If we have an environmental gradient in a sea with, let's say, the Baltic Sea, where you have a huge salinity gradient, you can search for genes that show signatures of adaptation to that gradient of salinity, for instance. And this is something that you can do by doing resequencing and mapping the reads against the host genomes. That then enables you to also associate within that genomic variation that you observe in space. It's also correlated with microbiomes, for instance. We could spend hours speaking about genomes, but as we don't have time, let's jump to microbial metagenomes, because it is as complex or more complex even. And it is when we go to microbial metagenomes, we are facing pretty much the same issue. So sometimes we do have a reference bacterial genome catalog that we can use to just map the reads that we have, sequencing reads we have generated against them, and create a reconstruction of the microbial community. And this is something that we can do, for instance, if we are analyzing Western human societies, if we are analyzing lab mice, etc. Those environments have been characterized so well that you don't really need to reconstruct new bacterial genomes, because most of them are well known. Of course, you have a chance to find something new, but the likelihood is lower and lower. But if we go to wild species, and most of you have listed wild species, tunas, swordfish, sea bass, the sea pen, the corals, all these guys have a lot of bacteria that are associated with them that no one have ever reconstructed the genomes from. And in those cases, it is extremely useful to use this approach, genome-resolved metagenomics, which basically is based on reconstructing bacterial genomes from metagenomic mixtures. You have genetic information of multiple bacteria all together, and using computational approaches, you try to cluster all this sequencing information into bacterial genomes. How is this done? So the first step is to do metagenomic assembly. And by doing metagenomic assembly, it means that the reads that are contiguous to each other, they are linked to each other to create what we call contigs, as we mentioned before in the genomes part. So what it used to be a lot of different short reads suddenly became much longer reads. Once we have this metagenomic assembly of contigs, the next step is to do what we call binning. So you need to get the different contigs, and you need to guess which contigs are coming from the same genome and which contigs are coming from a different genome. Also, which contigs belong to bacteria, which ones belong to host, to virus, etc. There are different approaches to do this, but we won't get into this unless there's a will to deepen into this, and then we might have some time for that. But the important point is that all this is done computationally. There are different attributes that are considered: the relative abundances of the mapping rates to the different contigs, the k-mer information, so how the genetic sequences, how similar or different they are between each other. And there are some other aspects that are used for guessing which contigs are coming from the same genome. And this way we create what used to be a metagenomic assembly with a different set of different contigs coming from different bacteria. We turn into what we call metagenome assembled genomes, or MAGs. And MAGs are incomplete, usually bacterial genomes, so they are not circularized. Bacterial genomes are nuclear, well, not nuclear, so the non-plasmid genomes are circular, the plasmids, most of them as well, but usually we don't, with using short read sequencing, we don't manage to circularize them. In order to create the entire circular genome, we usually need long read sequencing, PacBio sequencing or nanopore sequencing. Once we have that, then we are able to close the genomes. And the reason is that oftentimes we have genes that are duplicated. And once we have a duplication, if the reads are too short, the assemblers are unable to close that gap. And then they are unable to tell whether this fragment is attached to this one or it is attached to this one. So they are kept separately. If we have long-read sequencing, as the sequences are much longer, then we might be able to resolve this. The likelihood for resolving it is much larger. Once we have that, what we usually do is to assess whether the completeness and the contamination or redundancy of the genomes is good enough. So completeness refers of how complete the bacterial genome is expected to be. And this is based on an inference using single copy core genes. So most bacteria have a set of genes, a few genes that appear in most bacteria and appear only with a single copy. So they are not duplicated within the genomes. So using this information, if we are expecting 100 of these single copy core genes to be present in each and every genome, if we only recover 95, then we will say that the completeness of that genome is 95%. If we only recover 50 out of the 100 that we are expecting, then the completeness will be 50. The contamination assesses what is the redundancy of those. If, as I mentioned, these genes are expected to be found only once. If we found that gene twice or 3 times, that will increase the level of contamination or redundancy. And what we are interpreting with that is that we have included some contigs into that bacterial genome that most probably do not belong to that genome. It's some kind of an error that we have done, a computational error, for whatever reason. The completeness and contamination level at least enable us to see whether we relay on this bacteria or not. And we can plot this kind of figures where we have in the y-axis the contamination, and we want this to be as close as 0. And here in the x-axis we have the completeness, and we want to be as close as possible to 100. So our aim is always to have genomes in this edge here. Of course, when we reconstruct genomes from different samples, we get this kind of cloud of there are some bacteria like this one here has a 60% completeness and 5% contamination. Then how we deal with these ones to make them comparable to these other ones, it's a tricky question. And sometimes most of the people what do is just to set some threshold and say, okay, I won't consider anything that is less than 70% completeness and less than or more than 5% contamination. But still you have some variation here, that it is purely technical variation. And it is important to consider that whenever we are doing mostly functional analysis of the microbiome. If we are trying to predict what is the capability of a microbial community to do some kind of action, then we also need to consider whether the genomes that we have reconstructed are complete or not. Because anything that is less complete is more likely to have less capacities for doing metabolic functions. But it might be purely a technical bias or limitation. Because if that genome was complete, then it would have a larger capacity for doing that. So this is just something to consider. It's important to keep in mind what is the completeness and contamination, and then the actual strategy that is used to minimize the effect of the different completeness and contamination. It's something that is quite complex and it won't be covered in this lecture. Once we have reconstructed the different genomes, then we pretty much do the same as we did with the genome resequencing before. So we map all the reads that are from our samples, and then with this information, what we try to get is, for instance, the relative abundances of different bacterial taxa. And in this example, we have, most probably you have seen this kind of representations in which in the x-axis we have different samples, and in the y-axis we have the relative abundance of each, in this case, metagenome assembled genome, or MAG. So each tile here is a different MAG, it's a different genome, and then it is colored by the phylum in this case. So you can see here that in this case, these are cats, and the cats from Cabo Verde have a lot of this red. And the cats from Spain and Denmark don't have these reds or have very little amount of reds. So this way we can know whether there are taxonomic differences between different populations, different individuals, etc., etc. But the real power of genome resource metagenomics, of reconstructing the entire genomes or the near entire genomes of bacteria, is that we can do functional inferences. And this is the main difference that exists when we compare it to amplicon sequencing of the 16S RNA gene, for instance. That's the most common approach for doing microbiome research, because it is the most straightforward and it is the cheapest approach, but it gives you only taxonomic information. And sometimes if your taxa are not well reconstructed, so well represented, that can be also quite limited. There are ways to infer functional information from the amplicon sequencing data, and these are quite okay if we are talking about microbial communities that are well represented in the reference databases, like the human microbiome, like the mouse microbiome, could be also the cow microbiome. But if you go for bluefin tuna or swordfish or the corals, most probably the representation that you will get will be extremely biased. So that's the main advantage of doing genome resource metagenomics, that you are reconstructing your own bacterial genomes that you know you have found in your samples. And that gives us a lot of information. So here in this representation, each column is a different bacterial genome. Each column here in this heat map is a different bacterial genome. And these are colored here. It have been misplaced a little bit, but just imagine that this is a little bit to the left and it is aligned. This is the phylum of each one. So we have here the Bacillota A, Bacillota C, Bacillota, Bacteroidota are the brown ones that go to this chunk, etc. And then we have this phylogeny that we can build because we have entire genomes. So with entire genomes, we have really huge power to create rather robust phylogenies, much more robust than based on the 16S data. And this phylogenetic information is really relevant for doing evolutionary and ecological inferences. But on top of that, we also have this kind of complex heat map in which each row is a different functional trait. And in these functional traits, in this example, we have 6 traits that relate to the biosynthesis of nucleic acids. We have other 12 or 15 traits that are related to amino acid biosynthesis. So here, the darker each of these styles is, it means that that genome has a higher capacity for doing that metabolic action compared to those that have a brighter style. So for instance, you can see here, this column here is spanning about 8 bacteria. You can see that this is really bright. So there are very little blue here. This means that these bacteria here have far lower metabolic capacities compared to these other ones, the neighboring ones. They have so low that even they are unable to generate their own nucleic acids. And this is not an artifact. There are some bacteria that are unable to biosynthesize nucleic acids. So they need to get nucleic acids from the outside to replicate their DNA. And this happens in some specific microbial lineages, like this one, which is the RF32, that is called. So these are bacterial lineages that have been reconstructed, also have been discovered quite recently, within the last 10 years, and only using molecular methods, because these bacteria are so metabolically, let's say, I won't call it useless, but metabolically so limited that it is impossible or extremely difficult to culture them in the lab, because they have a lot of auxotrophies. You cannot just expose them to different nitrogen or carbon sources and they will replicate, they will just die. They have so many dependencies on other bacteria that generate some metabolites that then they can use for their own replication, that they have been completely overlooked until using these kind of approaches. And now we are seeing that this kind of animal, or this kind of bacteria are actually really widespread. And when we are talking about host-associated, animal-associated microbes, they are quite prevalent. So this is a representation of what is the power of using genome resource metagenomics. These guys were completely overlooked when doing culture-based analysis or amplicon sequencing analysis. And only now that we are doing genome resource metagenomics, that we are reconstructing bacterial genomes altogether, then we are able to surface this and try to understand what is the role in the microbial communities. So with genome-resolved metagenomics, we can answer these kind of questions like which bacteria are there, at which proportions are there, and most importantly, which functional capacities do they carry? What is the contribution they can make to the host, potentially if the diet is changed, if the temperature is changed, etc., etc. So this is what I wanted to tell you about the bioinformatics. Now we will jump into the statistics and we will cover that quite briefly. But before that, I would like to ask you again, how much of the explanations of this session you feel you have understood? And then, of course, if there is any question, but I have lost my faith now, so just let me know. And now I would like to insist, now that the points have dropped like 1.5 points, now I will insist until I hear a question. So please ask a question. Even if it's a dumb one. I want to continue until I hear a question. And don't do the trick now changing your question, to your answer to a higher score to avoid the questioning. Because it's 1/3 of the people don't feel that they have grasped with most of it. There must be a question about something. Please help me. It can be a question about your own project. It can be just take advantage that I am here. And if there is something that is related to what you are doing, I will be very happy to try give you a hand. I cannot believe Italians are so shy. I cannot really believe that. You don't leave me another chance than just asking the question myself. So who's the one working with the bluefin tuna? Is the plan to do any genomic analysis or microbial metagenomic analysis?
Sorry, can you repeat the question?
If the person who will be working or is working with bluefin tuna, whether they are planning to do any genomic analysis or microbial metagenomic analysis with it. You don't know? No. What about the one working on swordfish?
Oh, no, we didn't. We just used population genomics.
Okay, and how was the experience with the mapping? How did you feel that the quality of the data you got with the quality of the reference genome was good enough for your purposes?
Oh, yeah, the quality of the genome was almost good. The only thing is that we have to consider that the reference genome is one individual. So it's always a sort of bias when we search for SNPs.
Mm-hmm. So it is quite common that whenever you are starting to generate reference genomes, if you use multiple individuals from different populations, you suddenly realize that one species is more than one species, and that one reference genome perhaps is not as representative as what you were thinking it is. And this happens quite often in species that have wide geographic distributions, that even though we might not have recognized, we might be dealing with more than one species. So, because then the reason being that usually we just characterized one individual for the structural elements, and that can be quite limited. I have another question for the person working on the Desmophyllum coral. Yeah.
Yeah, here I am.
Are there any plans for genomic or metagenomic analysis?
For the metagenomic one, no, but for genomic, yes. We have to analyze DNA from two different populations, one from the Adriatic Sea and one from the Iranian Sea, and we want to analyze the population structure, but we didn't it yet, so we will do it in the next year, probably.
Mm-hmm. And what is your approach then? If you don't have a good quality reference genome, what are you planning to do?
In the NCBI, that was like just scaffold. So right now, I'm not thinking about it because I'm doing like another thing, but I will think about it probably right now because the time is running.
But there are alternatives. So whenever there is no reference genome, and for a long time, reference genomes have not been available. So this would be the gold standard for doing SNP-based analysis. But there are ways that you can use RAT-seq, or you can use other approaches that use a reduced representation approach for the analysis, where you are unable to span the entire genome, but at least you get enough information to get a genetic structure information and population structure information. So this is also something to assess. So if there is no reference genome available, then there are alternative methods that are worth exploring without relying always to the reference genomes, because sometimes it might be impossible to generate it, or it might be too complex, too expensive, but this is not a must. So this is not the only way to do it.
Thank you.
Cool. I'm so glad that I can hear your voices. So because I can see you're so tiny. So I don't know whether you're real either. So now I can see that's the case. So thank you. Are there any questions now? Come on, guys. I'm feeling bad. Okay, I hope the last one can trigger some questions. So we saw that the score of the understanding has dropped from 7.4 to 6.1. Perhaps in the next session, we will go it's a little bit lower, but I will try to avoid that. So I will go slower, and it's gonna be shorter. I have removed a few slides, so we can make it a little bit more palatable. Because statistics based on this kind of analysis might be quite complex, and so we will try to keep it as simple as we can. So we talk about univariate and multivariate modeling. So just to keep it simple. This is just an important consideration to make. And it's important because when we use whole genome sequencing or metagenome analysis that encompass multiple bacteria. Now, wait a second. I hope you can hear me now. My AirPods ran out of battery. So hopefully you can hear me. So whenever we are analyzing whole genomes or microbial metagenomes, we need to consider that we are not only working with one element at a time. When we work with genomes, we might be working with usually SNPs, so single nucleotide polymorphisms, that we might have millions that we are analyzing together. When we are working with bacteria, we might have different microbial taxa, or we might have different microbial functions, but we often have hundreds or even thousands of bacteria that are analyzed together. When we work with this kind of analysis, what we need to do is multivariate the statistics, because we have not a value that is the response variable, but we have a matrix. We have multiple variables that work together. So response variables that are together. Then whether we are trying to predict what the change of this system is responding to a single variable or multiple variables will define whether we are using univariable or multivariable analysis. And I just wanted to mention this because oftentimes univariate and multivariate is used as a synonym of univariable and multivariable statistics. And when we are working with genomic and metagenomic data, it is important to keep this quite clear, because we might have a univariable statistical approach, but it's multivariate, like in this case. This matrix might be a community of bacteria, but we are trying to reconstruct, we are trying to model what is the change of that community responding to a single variable, let's say temperature or salinity. So this is multivariate, but it's univariable. Here we might have just a single pathogen. Let's imagine that we just have presence or absence of a pathogen. And this is univariate. It's just one element that we are trying to predict. But we might have multiple variables that we are trying to analyze together to predict what is happening with that pathogen. So that would be univariate, but multivariable. So I just want to mention this to make sure that you get the concept of trying to reconstruct the response of multiple elements together as a response of one or multiple variables. And this is the differentiation. So we go again. We have a single value that could be pathogen, yes, no. Could be also genome A or genome B. Let's imagine it can be a species A or species B. We just have one thing that we are trying to say. It's a one yes, no response. In here, imagine we have 100 genomes of bacteria, and we have the relative abundances. We might have millions of SNPs that we might have different alleles in each of those. In this case, if we are trying to model how this change as a response of a single variable, then it would be a univariable, but univariate and multivariate. If we are trying to do the same but multiple variables, then will be multivariable. What we can do, and what we often do, is to condense multivariate information into a univariable space. So we might have a community of microbes, but if we obtain one statistics, could be Shannon diversity, for instance, of the community,
then suddenly what it used to be a multivariate element becomes a univariate value. So the entire community is represented by a single variable of diversity. Could be Shannon diversity, could be richness. We might have 100 different genomes, different bacteria with different representations. But the moment that we say, okay, what is the richness of that system? 100. Then we have just one value there that is representing the entire system. So it's not multivariate anymore. So there are ways to simplify the system, and not all the analysis needs to correspond to multivariate modeling. The most simple representation of this is when we analyze microbiomes, for instance, is alpha diversity versus beta diversity. When we do alpha diversity, we condense all the information within each sample into a single value. When we are doing beta diversity analysis, we usually consider the entire set of bacteria that we have there, and we consider them all together. So that's a multivariate analysis. And then alpha diversity and beta diversity can be modeled as a response to a single variable or multiple variables. Here we have an example from this example that I added previously as well. So these are cat microbiomes. These are intestinal microbes of domestic and feral cats. And here we have some univariate data points. And this each dot here is a different sample. And we have just a single value that we have represented the entire community as richness, how many bacteria are in the intestine. This neutral diversity is the Shannon diversity. This is the phylogenetic diversity, and this is the functional diversity. So we can condense all this information into single elements and then run rather simple modeling. We can use linear models, linear mixed models, but as a responding to a single element. And this is quite simple. And most of you, I guess, have done or are doing this kind of statistics where you have one, in this case, a continuous variable that you are trying to predict using one or multiple variables. In contrast, if we want to represent this kind of two-dimensional space, here we need to condense all the information of different species and their relative abundances in order to generate this kind of spatial aspect of how each bacterial community is represented in this space. And this is one of the simplest multivariate analysis that we can do. This is just for showing you that even though the community composition of the microbiome is multivariate in nature, we can condense that information to create simpler statistics and simpler modeling approaches. But it is also it is important to always consider that in essence, microbial communities and also genomic information in essence is extremely complex, because in our system, we might have multiple samples. Within each of those samples, we have multiple bacteria, for instance, or multiple genes. And inside of these bacteria, we might have multiple functions. So then the challenge comes on how to model all this together. But if we manage to do that, then we are able to generate really cool results that can help us understand a lot what is going on with our system. In this example from a recently published paper, what we did was to see which bacteria are associated with feral origin of the cats or domestic origin of the cats, and at the same time to see whether they are associated with aggressive behavior and tame behavior. So what we did was to collect animals that were coming from nature. So they were all cats that at some point were domestic cats, but some were gone feral. So it means that they don't live in association with humans anymore. And some other cats were house cats. So we characterized the microbiomes, but we also did some behavioral analysis with the cats. And we saw that there are some bacteria that not only associate with the feral or the domestic origin of the cat, but also their behavior. And you can see here by the colors can be seen quite clearly that you can see that all the pseudomonadota are linked to domestic cats, and a lot of them also to tame cats. So we know that the animals in domestic animals usually they are tamer. They don't attack the humans usually. Sometimes they do, but less often than the feral animals that are much more aggressive. And this can be also recapitulated from their microbiomes. But the most relevant thing and the most important thing with this is that we can do similar analysis at the functional level. As we do have all these traits that I showed previously with each of these bacteria, with all the traits, the functional traits, we can see whether there are any functional traits that are associated with the feral origin or the domestic origin, or the aggressive or the tame origin. And something similar could be done in your examples with could be the tunas that if you are comparing tunas from the Atlantic and the Mediterranean, or two seas and the Adriatic and the Tyrrhenian seas, whether you see that there are not only taxonomic differences between these two populations, but whether we can also find functional signatures that are associated with these changes of bacterial taxonomy. In this case, for instance, we saw that there are some functions that are linked to the feral origin, like the capacity to degrade threonine, the capacity to produce spermidine, the capacity to biosynthesize vitamin B12. They are associated with both the feral origin and also the aggressive behavior. So all these that are green are the ones that are matching the origin with the behavior. There are some other functions that were not showing the same signal in both cases, but there were many that they did. And using this information, then we can create this kind of ordinations. For instance, these are each dot here is one bacterium, and they are represented in this space based on their functional capacities. So the dots are the bacterial genomes. The closer those dots are, the more similar they are functionally to each other. What we saw is that when we have aggressive animals, they tend to have bacteria that are capable of producing more spermidine, degrading more threonine, biosynthesizing more vitamin B12, etc., etc. There is a displacement of the functional space of the microbiome. And this is something that you can do by modeling using multivariate statistics. There is if you are interested in this, I won't get into the details here, but there is a package that we use. It's called HMSC, which stands for Hierarchical Modeling of Species Communities. And it's a really robust and really cool package. It's an R package, and I think it's also available in Python, that you can do this kind of complex multivariate analysis that enables you to identify signatures of functional variation across different populations, timescales, spatial representation, etc., etc. And then you can see that if you plot each of these genomes, so we have all these dots are the bacteria that are associated with tame cats, while these other ones are associated with the aggressive cats, and we plot them based on their capacity for doing different metabolic processes. We can see that the animals that are associated with aggressiveness have one means that they have that capacity for doing that metabolic function. 0 is that they don't have a capacity. So we see that in terms of threonine degradation, we have a lot of genomes that are much more capable of doing that in the aggressive cats than in the tame cats. And the same happens with the proline biosynthesis, D-mannose degradation, etc., etc. And this is just to show you an example of how we can go far beyond just characterizing the microbial community with bacterial taxonomy, but we can actually get a grasp of what is the functional capabilities that all these microbial communities have, which could potentially explain why some bacteria, why some animals are behaving one way or the other way. But these analyses are just based on correlations. And this is something that we always need to keep in mind. So these were cats that we collected in the environment, in the wild, and then we characterize them. We can set this kind of associative analysis but we cannot really say whether there is a causal correlation between the microbiome changes and the changes in the behavior of the animals. In order to do that, we would need to go back to the second session and do experimental work in which we transplant the microbiome of cats from aggressive animals to the tame animals and see whether their behavior changes, for instance. So something similar can be also foreseen for different marine species that you might consider for your assignment of the grant proposal you will need to hunt over me in May. So this was closer, so it's shorter, but I am willing to extend on any of those aspects if you find them relevant. Before that, I would like to get your assessment of how much you understood. And we'll start from two extremes almost. So. So now we are around 7. Please ask a question. I'm otherwise I feel very alone here in my office. A question? Yeah. Is that a question?
Yeah, today. The cat study was done in different regions. Did you take into account when looking for the analysis?
Yes. So when we do this kind of analysis, we can include multiple covariates in this case, or we can also add random effects in the models. And in this case, the location was included as a random effect. So we could account for that information as well, because one of the largest effects, actually a larger effect than the origin or the behavior, was explained by the geographic region the cat was coming from. So we had to control for that. So in the modeling, you can do that. Yeah.
Okay. Thank you.
You're welcome. And thank you for the question.
Any other questions?
I guess no.
Okay, so we can continue. We are just we are finishing, so we will probably end ahead of time. But before that, I would like to remind you about the assignment that we should expect something that it's about 3-4 pages long, and it has to be delivered to me by May the 9th. If you do it earlier, better. It's absolutely fine. And remember, so this is the structure that they're expecting. So you will have access to the slides. So there's you don't need to take the notes now. But I would like to get a grant proposal that contains all these 6 sections and then deliver them using this form. Now, I hope that some of you, there is some question about this assignment. So I would really prefer to have that question asked now so that the response is given to everyone. rather than starting answering questions one by one to each of you. So if you have any questions about the assignment, please ask them now. You are the shyest Italians I have ever met. So. I have 2 Italians in my group and they don't stop talking. So
Maybe it's Monday morning.
Maybe.
that could be. That's also true. Perhaps Monday mornings they are not here very early.
Okay, question no? Okay.
No.
Thanks, Antton.
No.
Go.
So if there are no questions, I have one final slide. It's mostly for assessment of how the lecture went. So if there is nothing else, I would really like you to spend 2-3 minutes answering 2-3 questions, if that's possible.
Yeah, sure.
This is the assessment. I think it is private, so we won't see the responses on the go, I think. But if we do, there is no problem. But if you can join the Slido, and it's 3 questions about assessing the lecture, so we know whether it is useful or not, and it is worth doing it this way in the future or not. Because compared to 3 years ago, I changed it mainly to fit it to the assignment. Because 3 years ago, there was no need for handing over this assignment, but so the contents were slightly different. But it would be good to know whether you have learned anything, whether this is useful for you, and then we can discuss how to structure it for the future. Well, thanks a lot for providing the feedback. And so for those that are apologizing for not being very interactive, you are more than excused. It's not easy to make an interactive session with this setup, but I would say you were pretty active on Slido at least. So thanks a lot for that. So I think we can wrap up whenever you want. If you have any final questions, anything you would like to say, just take advantage of that. Otherwise, we can finish whenever you find it done.
No, I think there are no questions.
Thank you very much, Professor.
Thank you, and I hope you enjoy the teaching with the entire teaching week.
Thank you.
See you. Goodbye.
Thank you again.
Bye-bye.
Goodbye.